And so the reason why we're kind of differentiating these two spots on the promoter is because different things happen at the Tata box versus the initiation site. The Tata box is where some of the helpers of the RNA polymerase bind to. And the name of those helper proteins, they're called transcription factors. Right? So that's where, again, the transcription factors bind to the Tata box of the promoter region. And it turns out that there are several different types of transcription factors, and they all bind to that Tata box of the promoter. Again, the purpose of these transcription factors is that they kind of help the star of the show do their thing. And when we're talking about transcription, remember again that the star of the show, the enzyme that makes it all happen, is RNA polymerase. And where it binds to the promoter, but specifically it binds to this initiation site of the promoter. It turns out that in order for the RNA polymerase to bind, it has to be associated with a protein called the sigma factor. Right? So it turns out that RNA polymerase will only be able to find this initiation site if there's this sigma factor that's bound to it. Right? If this sigma factor was not bound to the RNA polymerase would never make its way towards the initiation site. So again, here's another protein that's helping the RNA polymerase do its job. It turns out that RNA polymerase needs a lot of help if you're talking about a eukaryotic cell in order to transcribe that gene. So there are usually several transcription factors that kind of glom on and make sure that the RNA polymerase stays attached to that initiation site. There's other helpers too. So there's another portion of the promoter that's called, so that's another transcription factor. A whole bunch of them are needed to help RNA polymerase. But again, it turns out that there's a lot of help that's needed. So there's another portion of the promoter that's called the regulatory site, and it's here that another helper protein binds to. So there's something that's called a regulatory protein that binds to the regulatory site. And again, the job of this regulatory protein, like all these other proteins, is that it plays a role in making sure that the RNA polymerase stays bound to the initiation site. Turns out that that's not it. The RNA polymerase in eukaryotes at least need even more help. So if you consider this region here, so there's a region in front of the promoter, a little bit upstream of the promoter, called an enhancer region and a silencer region. And it turns out that there are proteins that also bind here that help RNA polymerase do its job. And so the protein that binds here that helps RNA polymerase, it's called an activator protein. That's what A stands for. And so again, when you're talking about eukaryotic cells at least, transcription again requires RNA polymerase, but it'll only do its job if there's all of these proteins also bound. And basically all these proteins help the RNA polymerase do its thing, which is to go down that gene and transcribe it. And so remember that after it hits the terminator region, that's when it knows that it's found the end of the gene. That's when it comes off. And again, what the RNA polymerase does is that as it goes down that gene, it uses it in order to make, it reads it in order to make a messenger RNA molecule. And the messenger RNA molecule again goes to the ribosome to tell the cell how to make a particular type of protein. So kind of the take-home point here is that in eukaryotic cells, there are a lot of proteins that are required in order to help RNA polymerase bind to the promoter region. So we mentioned that there's these things called transcription factors, there are these things called regulatory proteins, and there are these things called activator proteins. Again, they all help the RNA polymerase transcribe a gene. You might be wondering, in this picture here, so check out this activator protein all the way over here. You might be wondering, how does a protein that binds all the way over here, how's that going to help RNA polymerase attach way up here? Right? It looks like there's a big disconnect between the activator protein and the RNA polymerase that binds to the promoter. how does this help something that binds all the way over here? Well, it turns out that DNA, it's kind of flexible, so it can bend. And because of that bending, that's what actually allows the activator protein to interact with the RNA polymerase and make sure that the RNA polymerase stays there. Here's what we're talking about. So again, here's the promoter region. Here's the RNA polymerase. We said that there's all kinds of things that help it stay attached, including something called an activator protein, which is way over here on this portion of the DNA. But what you're seeing here is that the DNA can actually bend, and it's because of that bending that's what allows the activator protein to actually be in the neighborhood of where the RNA polymerase is. And again, all of these proteins here basically help the RNA polymerase attach to the promoter and perform transcription. One last thing we'll mention, so basically that's, you have the whole story as far as what happens during transcription. So one last thing we'll mention before then we leave transcription and look at translation is this. So again, what we've just looked at is how does a cell read a DNA molecule in order to make a messenger RNA out of it. It's this messenger RNA then that's going to go to the ribosome in order to instruct the cell how to make a protein. It turns out though that in eukaryotic cells, things happen to this messenger RNA before it heads over to the ribosome. And one of the major things that happens to this messenger RNA is that things are added to it. So to one end of the messenger RNA is added just a string of guanines, and to the other end is added a string of adenines. And basically by adding the string of G's on one end and the string of A's on the other end, it makes it so that the messenger RNA, once it gets to the ribosome, it can stay attached there. Right? So these G's and these A's play a role in helping stabilize the interaction between this messenger RNA molecule and the ribosome after it gets there. Right, so again, the big picture is that the messenger RNA in eukaryotes, after it's been made, and after it's had the G's and then the A's added, right, so the G's is called just a poly-G cap. So poly means many. So a string of many G's are added to one end, and a string of many A's are added to the other. After that happens, then the messenger RNA ends up going to the ribosome to be translated. And so we'll take a look at how that translation happens next.